Hi everyone. So the title of my talk is Andrew Kaufman is wrong about COVID-19. So here's a disclaimer. The intention of this video is to provide scientific evidence that refutes the claims made by the psychiatrist Dr. Andrew Kaufman that the symptoms of COVID-19 are caused by host exosomes rather than a coronavirus. The views expressed in this video are not the opinions of the creator of the video. Please make your own conclusions after the video. So I'm going to address the main claims from two of his videos. The first one I found was titled, "Is COVID-19 really an exosome and not a virus?" And this was posted by Hype Galaxy on April 5th, 2020. And the second video is called Dr. Andrew Kaufman Exposing the COVID-19 Magic Trick, the Sleight of Hand that Transformed Society, posted by David Icke on April 21st, 2020. The second video I will only go briefly into, as you'll see the first one runs quite long. So video number one, here are the conclusions made by Andrew Kaufman, and I'm going to go through them one by one. So here's claim number one: respiratory illness is caused by an insult, toxic or otherwise, which cause production of exosomes. This can be because of poison, stress, infection, such as flu or pneumonia, or electromagnetic radiation, as he claims. And that people will express exosomes when they are healthy and sick, but this goes up when they are sick. So this first claim is true. Respiratory illness is caused by these things, and you could get production of exosomes. So that's true. But this is not the whole story. So really, the only reason exosomes are produced here is because they're always produced all the time, everywhere. So they're very small membrane bound like vesicles of endocytotic, which means inside the cell origin, that are secreted by most cells in culture. So that means when they're out of the body, and as well in the body. So they have a variety of different functions. And what you're looking at here, this is a host cell, and this is the plasma membrane. And this here, the MVE, is a multivesicular endosome. It sounds like a fancy name, but it just means a bubble within the cell that contains these little smaller bubbles. And what happens is this MVE, the multivesicular endosome, will move to the plasma membrane. It will fuse to it because they're both made of a phospholipid bilayer, basically just like a fatty layer. And then these little tiny vesicles will get released into the extracellular space, and these are the exosomes. And if you look at the structure of the exosome, it has the same bilayer as the cell itself, as well as the addition of some proteins and some contents inside, which I'm going to get into in more detail later. But essentially what you need to know is that the cells release these exosomes, and the point is that they contain waste, as well as signaling molecules for other cells or genetic materials for various reasons. But this is not necessarily increased when you're sick. It can also happen at many other healthy times in your body. Okay, they're really all over the place. So the next thing I've added here to the first claim is that he says PCR amplification can detect both. This is a problem because he has a complete misinterpretation of what PCR, exosomes, and viruses are. So PCR can detect all kinds of things, but not really in the way that Andrew Kaufman describes. So this leads into his second claim, and here's where I can start to demonstrate why this is true. So here's the second claim, that rt-pcr actually tests for host RNA from exosomes. Okay, so I posted a video online on April 21st titled David Icke is Wrong About COVID-19, Scientific Evidence from a Molecular Biology Perspective. I'm going to put this link in the description. Please feel free to watch it. I, in detail, will explain why this claim is incorrect, because David Icke also made this claim during an interview with Patrick Bet-David. However, I will briefly describe some of the reasons here, and I will show you why PCR tests are SARS coronavirus 2 specific. They do not recognize host RNA from exosomes. Okay, so here is evidence provided by Andrew Kaufman supporting his claim. He says that exosomes and viruses are similar in size. Exosomes and viruses both contain genetic material. rt-pcr randomly amplifies genetic material. And here he brings up out of nowhere I found it to be a painful analogy about a hat being transferred between people at a baseball game. The idea is that he's saying that this PCR can amplify any piece of genetic material, and it's hard to find the needle in the haystack. And this is entirely wrong. It's a very specific test, and I'm going to show you why. So his evidence for this claim is all correlation, and there is no discussion of how PCR actually works to detect RNA. So here, actually, he misinterprets an electron micrograph of what exosomes are, and I'm going to show you this right here.
Here on the left, this is an electron micrograph, or a picture from an electron microscope of exosomes. And you can see here an exosome budding out of the cell, and it's essentially a spherical or circular, this is a cross section, so this is like a slice of the tissue, where on the periphery there are like these kind of globular densities, or little dots or circles, right? And inside the cell here, where it says MVB, these are the same as MVEs. That's where all these exosomes are when they're inside the cell before they bud out.
All right, so here he's actually made a really big error, and I'm going to show you why. So this is the image he was talking about here, and what he was claiming is that this that I'm circling here is an exosome, and that this has budded out of the cell. And prior to budding out of the cell, it exists inside the cell as an MVB. And these here are the MVBs. Now, MVB stands for multivesicular body, and I'll explain why that's important in a minute. But essentially, this is the paper that he gets this from, and it's a bit concerning because I don't think he actually read the paper. This is one of the first Google searches of exosome to come up, so I'm pretty sure he just pulled it off of Google and didn't read the paper, because if he did read the paper, he would see that he's wrong about what he thinks an exosome is. So in the paper, they say that they're all roughly the same size, typically 40 to 100 nanometers in diameter. Okay? If you look at the scale here, this is 500 nanometer scale, that's this line here. And if you look at this, that would make this exosome about 300 or 400 nanometers in diameter. So that's not an exosome. Actually, what the exosomes are these, I've put 3 red dots here. Any one of these black circles, those little guys, that's the exosomes. And what this MVB stands for is multivesicular body, and those are the exosomes inside of these larger endosomes that I showed you before, the MVEs, or multivesicular endosomes, and those get released. So what you're seeing here, this is not an exosome. Those are the little exosomes, okay? So he actually does not recognize the exosomes in this image, and he's claiming that he's an expert on exosomes and that he knows that they're causing COVID-19. So my question to you would be, do you want to take information about exosomes from someone who doesn't even know what they look like in an electron micrograph? Okay, so this is very concerning. He already doesn't know what he's talking about. Now he starts to talk about PCR. So I'm going to show you a clip from him talking about PCR, and then I'm going to show you some videos or clips from my last video. Okay?
The most important thing is it's not actually testing for the virus itself, it's testing for a sequence of RNA. Now, that sequence of RNA may be present in a virus, but it also may be present in other things. So I'll give you an example, and I think I mentioned this before, but let's say we had a person that we wanted to identify.
Okay, I don't need to play any more there, but now I'm going to just show you, he basically said that the RNA present in the virus can also be detected in other things, okay? And this is where he's very wrong. You cannot detect viral RNA in a human genome. It's just not going to be there. They're very specific to each other, and I'm going to show you here how this works, the rt-pcr. So here is rt-qPCR, or reverse transcriptase quantitative polymerase chain reaction, and this is used to test or to detect RNA from biological samples, okay? So reverse transcription is the first step. This is where you use these special enzymes essentially to turn the RNA into DNA. And really what this is doing, it maintains the sequence of the RNA but now you're using a more stable molecule to do your analysis. So RNA is inherently unstable, and it's not a molecule you can actually expose to the types of environment, or at least the environmental conditions that are needed to do PCR. So by converting it to DNA, that's all you're doing, is being able to do this procedure. So really the way the procedure works is that you have a sequence of interest. This could be any sort of DNA, human, or the viral DNA, or any DNA here. And then what you do is you actually use a complementary probe that has fluorescent dyes on it. So this probe, these colors here are representing different nucleotides, and they only match up with the complementary nucleotide on the sequence of interest. So for example, this probe is not going to bind at another region on this sequence, nor is it going to bind to any other region on any other genome out there, because it's been designed in a way to only target this specific sequence of interest. Okay? As well, the primer has been designed with the same specificity. All right, so this primer here, essentially it's just needed to start this chain reaction where this enzyme, this polymerase, will now travel along the sequence of interest, adding in nucleotides. And what this is going to do is this is going to displace the probe and release these dyes, and one of these dyes is actually inhibiting the other from producing color. So when you break them apart, now you can actually get the dye being produced. And in this way, you'll be able to see not only if you have the sequence of interest present by sending the probe in, but you'll also be able to see how much of it there is depending on how many of these reactions take place. So this is the quantitative polymerase chain reaction using the reverse transcriptase. And this is a great way that biologists can actually identify very unique sequences, specific sequences of interest, okay? So when you're using this to detect SARS coronavirus 2, again, this is your viral sequence here. This is the whole genetic sequence here with all of the genes. And I'll just remind you, it's about 2,900 and 900 RNA bases. And this is kind of what the sequence looks like. And it goes again to 29,900. Within this code, there is a code for this RDRP. And this RDRP, the RNA-dependent RNA polymerase, what I mentioned to you before, this is what actually has these very specific sequences for the primers and the probes that they use for the assays to detect for SARS coronavirus 2. So the key take-home here is that these are very specific sequences only present in SARS coronavirus 2. They are not in humans. So if you send these probes into human lung fluid, if you're getting a positive result, it means that the virus is present, okay? You can't really get a false positive. Let's just say you can't get a false positive from these tests in the human samples, okay? So this is the probe here, and I'm gonna show you that I've actually copied this into this really cool tool. So you can see this is called a blast search, the NC National Center for Biotechnology Information blast search. And what this does is it takes the viral sequence and searches it through all of the biological data of all of the sequences out there and tells you if this aligns with any of those sequences in a complementary way. Okay, so again, this is CC, GT, CTG, and so on. So CC, GT, CG. So I've just copied and pasted this here, and now I'm going to run the blast search. So if this sequence actually matched up with anything in the human genome, you would see it in the blast search. You would see Homo sapiens or humans showing up in this search. So when we do this search, the results that we get should all be SARS coronavirus 2. So it'll show up in a second here. It's just searching the database right now. And I'll encourage you guys to play around with this. Go on YouTube, find videos that explain how this stuff works, because it's really fun. You could play around with it. You could find human genes, you could find other genes, and you can actually look and see where they match up with other genomes in the databases. So in this way, it's actually a lot of fun. It's actually taking a lot longer than it usually does. Normally this search is pretty quick. I might just try it again if it's not working oh, here we go. So if you look here at this sequence, and all of the results are severe acute respiratory syndrome coronavirus 2 isolate. these are isolated coronaviruses, complete genomes, and you're getting 100% coverage. So really you're getting 100% sequence alignment, okay? And this is just for, if you look here, there is nothing other than SARS coronavirus 2. So this is really proof that this sequence, when you stick it into human or onto lung fluid, you're not going to get a false positive. This is going to only produce a result when you have the coronavirus, the SARS-2 coronavirus 2 present in the fluid. All right, so I'll stop there. And really, so that should explain a lot of it to you, and I'll just sort of reiterate this point here with this diagram. So this is the sequence I was showing you that I ran through the BLAST search, and this is the exact design of the fluorescent probe used to first detect SARS coronavirus 2. And really, if this is the SARS coronavirus 2 genetic sequence, you can see when you match them up together that this will sort of bind to the other one almost in like a zipper formation. Okay, and this is where the specificity of this test comes in. So this is the binding here is non-covalent binding between complementary nucleotides, and what this means here, as you can see in the diagram, the C's and the G's, the cytosines and the guanines, can bind to each other with these 3 non-covalent bonds, and the adenines and thymines can bond to each other with these 2 non-covalent bonds. So these are all aligned in such a way that they won't generally bind with each other. Let's just say they won't. So if you actually get a positive result, that means you have sequence alignment. This probe is not going to bind if there's 1 or 2 matching nucleotides. You need the whole thing. So if you put this probe into a human lung sample from lung fluid, and the lung fluid does not contain the virus, let's say it contains exosome genetic sequence, you're not going to get any alignment, and so you're going to get a negative result from the test. The probe that's used is so specific, it is not going to amplify exosome genetic sequences. This just simply won't happen. So really, Andrew Kaufman also talks about amplifying the negative results. I'm not going to get too much into detail about how PCR works, how the cycles work, but essentially you take your gene of interest that you've amplified or that you've targeted with the probe, and you amplify it. But the way this works is that this here, these 2 red lines, that represents what you're seeing here when they match up. So if they don't match up, you don't get any of this amplification happening. So really, the PCR is only amplifying positive results and not negative ones. Okay, so you're not going to get a whole bunch of false positives with this test. So really, I've proven this statement wrong. rt-qPCR probes and primers are designed to target viral-specific genomic sequences. rt-qPCR can distinguish between viral and human genetic material. rt-qPCR does not detect host RNA from exosomes when it's designed to target viral sequences. I should have added that in here, but when it's designed to target the virus, that's what it's going to target. Okay, so an example I want to give you is forensic science. So we've all heard of DNA analysis, and it can be used to convict people of crimes. So if somebody's not at a crime scene but they find their DNA there, this is often very, very compelling evidence that they committed the crime. They're not finding their whole body. They're not isolating the whole human at the crime scene. They're finding the DNA, and because DNA is like a fingerprint, even better, and it's so specific to each individual, this will hold up in a court of law. And in the same way, the scientific community allows or it has no choice but to accept RNA and DNA genomic analysis as holding up as evidence that something is present. I mean, it's the same as what works for us in the court of law. So really, Andrew Kaufman doesn't give enough credit to the power of genetic sequencing with the technology we have today. So his third claim is that COVID-19 is actually exosomes. So he believes that the result is not a cause of, that this is the result and it's not the cause of the illness. So he thinks that basically the exosomes are helping to remove toxins, but essentially that they are what's being picked up in the analysis. They're not actually detecting. So these people are getting sick from another toxin or poison, and they're using the exosomes and saying, we have a virus, but really they're detecting exosomes. Okay, so he's saying the virus does not exist. So this is a very bold claim, And he claims exosomes and viral particles are microscopically indistinguishable from each other. So this is, excuse the typo here, from each other. So this is actually his only piece of evidence. He just says that we're looking at particles from the lung fluid, and those particles, we think they're viral particles, but they're exosomes. Then we do rt-pcr on them, and we say that we have viral DNA or RNA, but really we have exosome RNA. So I've shown you why the RNA argument doesn't hold up, and now I'm going to show you why visually you can distinguish them from each other when you use a microscope. And really the problem here is that Andrew Kaufman really contradicts himself here because he demands better evidence from scientific papers, but he provides zero himself. He has nothing to back up his claims other than some weak correlations. So here I'm going to show you exosome membranes versus viral capsids. So here is what an exosome looks like, and you have the exosomes budding, as I showed you from the previous image. They're budding from the plasma membrane of the cell, and I'm going to show you a bit more about the composition. So there's a lipid bilayer that exists on the cell as well as the surface of the exosome, and there are surface proteins as well as internal contents like genetic material, proteins, and waste. Now if you look at a viral capsid, here's the viral particle, and it looks entirely different than an exosome. And what I really want to point out here is these spike glycoproteins. So these spikes exist on coronaviruses, and these spikes are very visible when you look at them in an electron micrograph, and they are not present on exosomes. So even though many of these can be similar size inside the cell, they look very different when you look at them under a microscope. So here, this is an image of a screenshot from Andrew Kaufman's video where he's showing that this is images that he claims are from this paper called A Novel Coronavirus from Patients with Pneumonia in China from the New England Journal of Medicine. And what he says is that this is showing the difference between exosomes and viral particles. Alright, so here, actually what he's showing, I'm not going to pull up the video, but because it's very long-winded, but what he's claiming is that these particles on the left and these particles on the right, one of them is a viral particle and one of them is an exosome, and that you can't tell them apart. However, here's the concern with this. This is actually evidence that he's manipulated data. Okay, and I'm going to show you exactly why in this next slide. So the original image that he got is supposed to be from this paper, A Novel Coronavirus from Patients with Pneumonia in China, 2019, and this is the only reference he has on this page. But if you look at the actual image from the paper itself, this is what it looks like. So on the left here, you could see the image of the viral particles here, and I'll explain soon why you can actually tell those are viral particles. And then on the right is a completely different image of these lung cells that have been infected by the virus. Okay, and so really what he's done is he's taken this image, he's added a second image next to it with no reference, and claims that these actually look indistinguishable from each other. Okay, and one of them is an exosome, one is a virus. So the one on the left here is the virus, as you can see by these spike proteins, which I'll explain more about later. But really, this is really bad. This is he is presenting false data. We don't even know if these are exosomes. These could be anything because he hasn't given the reference. So he hasn't given a reference for the exosome paper, and he's really manipulating information to fit his argument. He literally just pulled up this random image to try to say, look, these two things are indistinguishable from each other. So, I mean, in this way, this is so non-scientific, I'm really questioning his credibility here. So now if you actually compare viral particles to exosomes, here are coronavirus particles on the left. This is a Wikipedia image on the bottom. It's a very nice image, so you could find the reference through Wikipedia. And this here is the image that he presented from the New England Journal of Medicine. And really what I want to point out here is that these spike glycoproteins that I pointed out earlier, they're very visible on these viral particles. This is how you distinguish them, okay? So you could see them here. These are the spike proteins, all right? Now these would not be present on exosomes at all, period. And you could see the spike proteins here as well. So these are viral particles, okay? If you actually look at exosomes, this is the image he has here on the bottom with his wrong interpretation. Those are way too small to see. But if you look at a real image of exosomes here, you can see, so this is 100 nanometers, that's 100 nanometers. You're looking at things that are about the same size. There are no spikes on these exosomes, okay? Now, exosomes can have some differences in the way they look, but they will never look like this. They do not have spikes like this. Typically, you see a very smooth membrane. You don't see the protein sticking out of them at all because they don't stick out. They rest in the surface. Very different than these spike proteins. All right, so in this way, he's completely misinterpreted the data, and he's saying that you can't distinguish between these two, but even an untrained eye, I think, can say that these exosomes look quite different than these viral particles, okay? Yeah, these have no spikes. So really, I'm going to show you another example where he further manipulates data. All right, so here is a table that he presents during his talk, and this table, don't get me started on this table, none of it is correct. So he first talks about the diameter inside of the cell of exosomes, and he calls them MVE. I don't think he knows that this is multivesicular endosome, and I don't think that he knows that the exosomes are not the multivesicular endosome, as he's claiming here, but they are these little exosomes are within the multivesicular endosome, hence multivesicular. They have multiple vesicles in them that are going to be released as exosomes, okay? Once they're outside of the cell, you call them exosomes. So if you, really he's saying that this whole structure is an exosome, and then when it leaves the cell, so the diameter inside the cell is 500, the diameter outside the cell is 100. It makes absolutely no sense. And the reason why he makes the same mistake with COVID-19, this should say SARS coronavirus 2, but I understand he doesn't believe it exists. So the reason he makes this claim is because he's mistaking the endosome, again, for the virus within the cell. It's really, the endosome is a separate entity that carries the virus to the outside of the cell. So this information is entirely wrong. And as well, the rest of the table is also wrong. I'll just briefly explain. ACE2 is the receptor for SARS coronavirus 2 to attach to a host cell. Exosomes can have ACE2 receptors. They can also have thousands of other receptors. So there's absolutely nothing specific about ACE2 and exosomes. As well, exosomes, it says here, contain RNA. Exosomes can also contain various types of proteins as well as DNA or other materials, okay? So many different things can be inside of exosomes, not just RNA. And they say that exosomes are found in your lung fluid as well as COVID-19. Exosomes are found almost everywhere in your body, and COVID-19, as I have this reference here, SARS-CoV-2, it's also found in a variety of other tissues that are expressing this ACE2 receptor. So really this whole table is junk. He just threw it together. It's not referenced. I would just ignore this completely and further question his credibility at this point. So really, for this third claim, he has no evidence, and he's making the claim without real evidence. So my question to you is that, should we believe someone who doesn't know what exosomes are to talk about exosomes, and also manipulate scientific data? So what I would say to Andrew Kaufman is, if you do know so much and you're such an expert, please perform the necessary experiments to prove you are correct, like other scientists do, and then you can publish your results in a peer-reviewed journal to provide real evidence that exosomes are responsible for producing the symptoms of COVID-19. So he requires other scientists to give him amazing information that's perfect every time. I'm just asking him to produce something because he hasn't given us anything. So I'm just going to give you a snapshot. As you can see, that went on for quite a long time. His second video, after I've shown you what I just shown you, hopefully you understand he's not credible to talk about coronavirus at all. And especially the second video, I have like 5 pages of notes on this video discrediting him, but it's way too much to put almost even into a second video. But really, these are the claims he makes. He's talking about Koch's postulates as well as Rivers' criteria and whether it's met or not. He talks about SARS coronavirus 1 from 2003, as well he talks about SARS coronavirus 2. So he's claiming here that there's a lot of rumors and lies placed on this pandemic or this COVID-19 because there isn't any proof from these postulates. And Really, I described this in my previous video, but I'll mention it again here. The issue is he's jumping the gun way too quick. These tests take a lot of time, and essentially in 2003 or 2002 to 2004, when the first SARS coronavirus 1 was around, it took about 7 months to fulfill Koch's postulates, and it's only been about 4 or 5 months since SARS coronavirus 2 was first reported. And the reason this is because you have to do some very extended experiments. You do them with monkeys, you have to infect the animals, re-isolate the virus, reinfect new animals, a lot of regulations to go through. I'm sure they're sped up in this pandemic era. Either way, it takes a long time, and there's a lot of complicated antibody tests to do as well. But I actually pulled up some preprints that these tests have been done. So this is going to completely prove Andrew Kaufman wrong. And really, these are the experiments that I said needed to be done, and everyone knows in the scientific community that it takes time. And here they've shown that they've fulfilled Koch's postulates, and this is under review. So once this goes through peer review, they're going to clean up the paper and they'll be able to publish this. So you could look it up yourself. They've isolated this from the animals and reinfected other animals, showing that this is the causative agent for the disease. The SARS coronavirus 2 is causing COVID-19. Okay, and as well, there's another paper in preprint which is looking at the Rivers criteria, and here they're demonstrating specific immunity. So they did a lot of antibody tests here. Okay, you could look at the paper yourself, or I could discuss it more in another video. But essentially, these are my concerns about Andrew Kaufman's claims. First of all, he does not address the power of whole genome sequencing to identify viruses from known families. So he's just simply wrong here. He doesn't understand forensics. He doesn't understand any of that. He does not interpret rt-pcr correctly at all. So as I explained previously, he's completely wrong about this. He also manipulates data to fit his opinion. I think this is an egregious error. I don't think anyone should be allowed to get away with this. And his opinions at that point are also based on no evidence or even very weak correlations. So he doesn't show any causation at all with what he's talking about. And simply, I cannot trust someone who is uninformed on COVID-19 as well as basic biological concepts. I'm going to get my information from real scientists and doctors here about this disease. So my final thoughts: please research these topics on your own without blindly following what Andrew Kaufman or anyone else claims to know, including David Icke or myself or anybody. Look stuff up yourself, and this allows you to think freely and make an informed decision about the information you choose to believe. So thank you very much. If you liked the video, please click a thumbs up and feel free to subscribe to my channel. Thanks a lot.